Hello, and welcome to the Osteoarthritis Action Alliance Lunch and Learn webinar for September 16th, 2020. Thank you so much for joining us this month. Our presenter today is Duncan Lascelles. Dr. Lascelles is professor in small animal surgery and professor of comparative and translational pain research at North Carolina State University. His research program, Translational Research in Pain, develops methods to measure pain associated with spontaneous disease in animals and seeks to understand the underlying neurobiology. His work improves pain control in companion animals and facilitates analgesic development in human medicine through proof of concept studies in companion animals and discovery of relevant mechanisms involved in specific, naturally occurring painful disease states. Dr. Lascelles is director of the Comparative Pain Research and Education Center. He has authored over 200 peer-reviewed research papers and reviews, over 250 research abstracts, and over 30 book chapters. He consults for both human and veterinary pharmaceutical companies in relation to therapeutic development. He is also co-founder of Innovate, a company dedicated to developing innovative methods of measuring pain in animals. We are delighted that Dr. Lascelles could join us today, and his presentation is titled, OA Pain Research, Translational Scientists' New Best Friends. Dr. Lascelles, welcome, and I'll let you take it from here.
Well, thank you. So good afternoon, everyone. It is an absolute pleasure and honor to be talking to you as part of the Osteoarthritis Action Alliance lunchtime webinar series. You've probably got the gist that I'm going to be talking about pet animals, or at least non-rodent animals and non-human animals, because I am a veterinarian. And as you heard from the introduction, a lot of my research is based around measuring pain in companion animals. And that really is reflected in the title, Translational Scientists' New Best Friends. And hopefully, you know, the message here will unfold as I go through this seminar. And it's all my approach here. It's a little provocative. I certainly, you know, I'm not saying that what I do, what other people do, is the be-all and end-all of the answer to translational research, but it's just an additional idea, an idea that I think will enrich the translational pain research paradigm. So it all starts with this. Translational pain research is not producing new analgesics. And I think that all of us in pain research, translational pain research, need to take to heart, not to get too depressed about it, but just to say, okay, if the translational pain research paradigm is not working, then we need to reevaluate components of it. And just to underscore what you see on the slide there, pretty much in the last 25 years, there have been no new analgesics for musculoskeletal pain. Musculoskeletal pain is the mainstay of the burden of chronic or persistent pain in the United States. And so for 25 years, there have been no new analgesics that have broad applicability. And you may say, oh, we've seen new analgesics come onto the shelf. Yes, you have. You've seen combinations of non-steroidals and opioids and other already known analgesics. There have been no new analgesics for 25 years. So pain translational research is not working. And so the question then comes, well, where does translational pain research go wrong? And the process from drug discovery, preclinical research, through clinical trials, and the FDA process of review and approval, many components could be criticized, have been criticized, and certainly could be tweaked, changed, or altered to improve the process. I want to focus on the preclinical component here, and there has been a lot written about the problem of a lack of translation of the exponentially increasing amount of basic science research that is going on, the lack of translation of that into new therapeutics for humans. And so names here that I'm sure you'll recognize: Jeff Moe, Larzan, Nielsen, Andrew Rice, Natalie Piercy-Doucet. I mean, a number of people here, prominent figures in pain research, discussing and talking about why this exponentially increasing amount of basic pain research, with so much being learned about mechanisms, has not translated into new therapeutics. So how I'm going to approach this is, instead of saying, well, you know, dwelling on where does preclinical translational pain research go wrong, I'm going to turn this around and say, where could preclinical translational research benefit? And I'm just going to look at 3 areas: enhancements to models, enhancements to outcome measures, making them relevant to the problems that people experience, and the relevance of the target. Starting with models, and these are just a few slides to kind of set the scene here. So starting with models, I think one has to ask whether, when I inject complete Freud's adjuvant into a rat paw, and this is my work here I'm showing with the rodent, I am injecting complete Freud's adjuvant into a rat paw, and I do that as a model of persistent pain as part of my osteoarthritis research. When I do that, is that really reflective of what you see on the right of the screen? An older gentleman there with knee osteoarthritis pain, who has had that pain for decades. And so I'm not going to answer this question, but I think we have to ask the question, are the models reflective of the target disease condition? Because we base so much on the models and on the results from these models. Likewise, outcome measures. So the standard, for anyone involved in basic pain research, the standard, the first way that you assess pain, in inverted commas, is by testing sensitivity using a von Frey device, electronic von Frey or von Frey hairs. And you can see in the picture there, the rat has just lifted its paw in response to the von Frey device being actuated from below the paw up onto the paw, and the rat has pulled away. And just recently, there was a really nice article in Nature by Huang et al. Showing quite clearly that that pathway that the rat is using for paw withdrawal is probably quite separate from the pathway that encodes the affective component pain, which is the feeling of pain. It's the feeling of pain which is adversive. That's what we want to try and treat in people who want to decrease that aversive feeling of pain. So the primary outcome measure here in basic science are often these reflexive measures, and they may not be reflective of the adversive, the affective components of pain. Then there's the relevance of the target. So in basic pain research, and this on the right of the screen, you'll see this is a TRPV1 expressing neuron, so it's essentially a pain-sensing fiber. This is some work by a colleague of mine, Santosh Mishra, where he basically characterized all the receptors that are on these TRPV1 expressing neurons. Now, as a pain researcher, you would look at that and think, well, this is Christmas, this is holiday time. I mean, I've got so many receptors that I can develop therapeutics against. And that's the positive side. The downside is there are so many receptors, which means there's got to be redundancy in the system. And so if you develop a specific blocker to one of these receptors, you're only going to see a clinical benefit if that particular target is of relevance. So in basic science, we have a much greater understanding of the phenotype of these sensory nerves. We understand what receptors are expressed. And sometimes I think we jump to developing antagonists or blockers, drugs, therapeutics that will interact with specific receptors without asking the question, is that receptor of clinical relevance in the target disease? So I've outlined sort of 3 areas of problems there. And so here's my proposition: with naturally occurring painful disease may enhance the translational pain research paradigm. Why do I think that they may be able to play a role? Well, because these naturally occurring painful disease models, so now I'm talking about people's pets with naturally occurring painful disease as a model, they may better reflect the complex genetic, environmental, temporal, and physiological influences present in humans. So it's almost like a stepping stone between basic rodent studies with induced models but between that and human clinical trials. And so this is an image here just showing sort of discovery, and then you've got this point here where you jump into human clinical trials, phase 1, 1A, 1B, phase 2, 3. And what I'm going to talk about for a little bit now is how pets can play a role at this point here as a verification bridge to inform the critical go, no-go decision-making point. And then also in discovery of the target, starting with this verification bridge. And what I mean here is, if any of you have dipped into drug development, therapeutic development, you'll know that prior to starting human clinical trials, there are some hard questions to be answered. Which of the candidates are we going to move into clinical trials? Really important question, because that's when you really start to burn through the cash. That's when drug development becomes very expensive. And so what I'm suggesting here are that trials in pets with naturally occurring painful disease could help inform that go, no-go decision, could help inform which therapeutic to move forward. And I'm going to, this is the talk around OA. So my example here is long-standing OA pain. Firstly, if we're going to use pets as that to help inform that go, no-go decision-making process, then the model, the condition in pets needs to have fidelity with the human condition. And for osteoarthritis, biomechanically, structurally, histologically, genomically, and molecularly, human, canine, and also feline and equine OA are very similar. You'll see two radiographs here. The one on the left is a human coxofemoral joint. The one on the right is a canine coxofemoral joint. The disease is extremely similar, and the disease can be associated with pain in a very similar way in dogs as it is in people. The other thing here, I think, which is worth thinking about is, does the disease look like the human condition? So just watch my father here. So he had multiple joint osteoarthritis when he was alive. You can see how he struggles to move. And then watch this dog here. This dog has multiple joint osteoarthritis. And I think when you look at those two, you can't help but see the similarities. So now, not just molecularly and structurally are the diseases similar, but also in the look of the whole animal. And I think this dog is much more similar to my father and others with osteoarthritis pain than the rats, where I induce discomfort by injecting compounds into the paw. Now, so the models, I think, have fidelity. The naturally occurring painful osteoarthritis in pets has fidelity with the human condition. But these models are only going to be useful if we can, one, measure pain, and ideally, if we can measure outcome measures that are relevant to humans. So here is a little schematic showing how chronic pain impacts multiple dimensions. And all of these are interrelated. So chronic pain impacts gait and movement. It impacts one's ability to perform the activities of daily living, so function. It impacts sleep, cognitive function, social relationships. It alters how sensitive you are to environmental stimuli. And so these are dimensions that are impacted by chronic pain in people. And so which ones of these can we measure in dogs with osteoarthritis? Well, we can measure gait and movement. We can measure function, the ability to perform activities of daily living. We can measure sleep quality, cognitive function, affective states, sensitivity. So we actually measure, and we're really good at measuring these dimensions. All of these dimensions really reflect the problems that people with osteoarthritis pain face. Just as an example here, I would, arguably, I would say that we are much better at measuring limb use and gait in dogs than we are measuring limb use in rodents. Our ability to measure limb use is on a par with the measurement of limb use and gait in people. And here you see a dog just standing on a pressure sensitive mat, and we're recording the amount of body weight that is being put through each individual limb as a measure of spontaneous joint pain. And then here we have measurement of kinetic values in a dog being trotted across a force plate. We can also measure activity, and I've actually got a lot of interest in measuring the quality of motion in relation to joint pain. And we then use, so these are little activity monitors mounted on dogs and cats. We use these data to assess novel therapeutics. Here we're assessing an anti-NGF monoclonal antibody, osteoarthritis pain in cats. And here we were assessing the sleep quality and the effects of a non-steroidal on sleep quality in dogs with osteoarthritis. So these proof of concept studies then, because the model has fidelity with the human condition, that is osteoarthritis pain in dogs, has fidelity with the human condition, we can measure outcomes, we can therefore run these proof of concept studies. And you might well ask, well, does this work? What is the predictability of a proof of concept study in companion animals? How well does efficacy of therapeutic in a dog with osteoarthritis pain predict efficacy in humans? This table here speaks to that. It's a small table at the moment, but if you look down here in the left-hand column, we've got the drugs, the therapeutics, and then each column here subsequently shows efficacy in rodents, efficacy in dogs, and efficacy in humans. And what you can see as you scan down this table is that everything works in rodents because everything has been developed in rodents, in these rodent models. But where there is efficacy in dogs, you see efficacy in humans. Where there's not efficacy in dogs, or it's unpredictable efficacy, it's not consistent efficacy, you don't see efficacy in humans. So again, so as an example here, the NK1 antagonists, no efficacy in dogs with osteoarthritis pain, no meaningful efficacy in humans. So this table is looking pretty consistent here, where there's efficacy in dogs, there's efficacy in humans, meaning that these proof of concept studies run in pet animals can probably predict efficacy in humans. The sites and expertise are available. These are pet animals, these are members of the household. These studies must include veterinary oversight and must have ethical oversight and be approved by local IACUC committees and have fully informed owner consent. So they have to be run sensitively and well to a high standard. But they can be run, the sites and expertise are available, and essentially these studies can be run exactly the same way as human clinical trials are run, whether it's parallel group, randomized blinded studies, whether it's crossover studies, adaptive design studies, the same study designs can be recapitulated in these naturally occurring pet models. And it's being done. This is a recent publication, actually from Eli Lilly, who ran a study in pet dogs to determine which of 3 candidates for OA pain to move forward into human clinical trials. So this is something that is not just a concept, is very much up and running and ongoing. I want to end here just by briefly talking about discovery. So going back to my earlier comments, there are a lot of potential targets that we can interact with in the pain transmission pathway, but a target, our therapeutic is only going to show clinical benefit if the target is playing a predominant role in the generation of pain. And not all targets are playing important roles in in the generation of pain. So we have this buffet of targets that we can choose from, but the question, we have to ask the question, is this target actually relevant in the target disease condition that we are looking to treat? So to address this, if you think about what I've just said, companion animals can be pain phenotype. That is, I can measure very accurately the way that pain in a particular pet animal is impacting multiple dimensions. So I can phenotype these animals. Veterinarians have unprecedented access to biological samples, much fewer controls than on the human side, and pet animals get euthanized. And so we have now the ability to phenotype animals that are potentially undergoing surgery, and we have the ability to phenotype animals that may be euthanized. It gives us unprecedented access to biological samples with associated metadata on the pain status. So now we can take these tissue samples and we can interrogate them. We can look at targets of interest, or we can interrogate them in an unbiased way, RNA-seq, the omics approach of your choice. Interrogate them in an unbiased way and ask the question, is the particular target we're interested in of relevance? Is it altered in the naturally occurring painful state? Or we can say, what is altered in the naturally occurring painful state, and make that then the target of interest to go back into preclinical rodent work to understand more about the mechanisms involved. And these pictures here just demonstrate, this is a dog undergoing surgery. We can collect joint tissues at the time of surgery. This is a dog that was euthanized here, and I've done a total dorsal laminectomy, and I'm retrieving dorsal root ganglia here, which we then can evaluate through various standard molecular techniques to understand what is altered in the naturally occurring pain state. And we've been working on this over the last few years. We've been collecting tissue samples, we've been collecting biological fluids, and we did exactly that. We interrogated those samples, those tissues, in an unbiased way, and discovered that a hitherto kind of unrecognized signaling axis appears to play a very predominant role in osteoarthritis pain. That is the interaction of artemin, a neurotrophic factor, with GFR alpha 3, its receptor. So this is leading us to go back into rodents to better understand this signaling mechanism and how best to interact with it. But I guess the important point here is the starting point is that we believe this signaling pathway is playing a predominant role in osteoarthritis pain in dogs, because that's where we started. And in fact, since we did this work, it's come to light that other people have been looking at human tissues and also found the same thing. So to conclude, companion animal models, so I'm talking about pets, people's pets, with naturally occurring painful disease, these companion animal models, I believe, can contribute to translational pain research in two basic ways: acting as a verification bridge between rodent preclinical and human clinical studies, essentially testing drugs for efficacy prior to human clinical studies. And also, they can contribute to discovery or understanding the face validity of targets. Tissue from naturally occurring disease states may provide vital information about the neurobiology of pain in the natural disease state. And again, these two conclusions here rest on the fact that for many diseases, and osteoarthritis pain is one in particular, for many diseases in companion animals, that disease is extremely similar to the human condition. So I'm going to stop there and be very happy to take any questions.
Thank you, Dr. Lascelles. That was really, really fascinating. As an animal lover, it was just interesting to hear the relationship between, what can be used for treatment in animals and transferring that over to humans, but also just in the idea of maybe even improving quality of life in our pets. I did have a question for you. You talked a little bit about the predictability of efficacy in treatments in dogs and humans as compared to rodents. And I wonder if you have come across any treatments, whether that's pharmacologic or non-pharmacologic supplements that maybe are more effective in pets than they are in humans.
That's a really good question. So honestly, I focused on trying to understand, if there is efficacy in pets, in dogs, do you see efficacy in humans? So I've been trying to understand that matchup. I think we are starting to see a few treatments where there may be more efficacy in pets than humans, and one of those is the EP4 receptor and antagonist. So this is acting further down the cyclooxygenase pathway. We actually have, in veterinary medicine, we have an approved EP4 receptor antagonist in clinical use across the globe. And I believe that on the human side, there have been they have struggled to measure efficacy of these EP4 receptor antagonists. It's a great question, and I think in the future we may see exactly that. We may see some therapeutics that have more efficacy in pets than humans.
Question here: how broadly has the pharmaceutical world embraced this concept?
Yeah, that is a great question. Pretty broadly, but rather quietly. So let me just explain that. The major pharmaceutical companies, so Pfizer, Regeneron, Eli Lilly, Amgen, they have for a number of years been very interested in this concept, and many of them have quietly started studies basically using this concept. And I say quietly because they have felt, oh, this might give us an edge on the competition. So it's not as if they're all talking to each other about this. And then what's been interesting recently is I've really seen a change in the NIH. The NIH has now embraced this concept as a way of validating therapeutic targets. And in fact, I just had a R01 funded doing exactly that, using, as well as rodents, using pet dogs to validate particular therapeutic targets. So it's certainly something that's becoming much more mainstream, onto the main stage. I would like to emphasize, though, that this is not a matter of throwing the baby out with the bathwater. This is not a replacement for rodent studies. This is and I think I mentioned this at the start, this is the way to enrich the translational pain research paradigm. We're basically pulling information from more directions to better understand the potential of particular therapeutics.
That's great, thank you. Are there any other questions from the audience? Feel free to type them into the chat box. And Dr. Lascelles, I just a personal note of curiosity. So you've talked a lot about targeted therapies and analgesics and pharmaceuticals, but I do wonder how much your clinic gets into kind of weight management and kind of the non-pharmacologic treatments for pets.
That's a good question. This talk was really based on around pharmaceutical development and drug development. We actually run trials. We run as many trials looking at non-pharmaceutical therapies as we do pharmaceutical or biologic therapies. So we actually have an ongoing study looking at a supplement, ongoing study looking at weight management, an ongoing study looking at manual therapy. You know, if this talk was based on those, you'd find the same thing. It's the therapies that appear to be beneficial in dogs are shown to be beneficial in humans, and vice versa. Those that where we can't detect efficacy in dogs don't appear to have that consistent signal of efficacy in humans. And so I think, you know, it's actually going to be a really interesting way to assess supplements, which is a very robust market, completely unregulated. And I think it would be good to better understand the value of some of these supplements that are purported to have benefits in patients with OA.
It certainly seems to be an area that's of interest to patients. Often a question that comes up about, you know, what supplements they can use, yet many of the recommendations are kind of staying away from supplement use
Right,
this time.
right. Yeah, yeah.
Well, thank you so much for being here today, Dr. Lascelles. It was a really fascinating presentation, and we're so grateful for your time and expertise. So that does conclude our presentation today. I will be sharing a poll here in just a moment. If you have time to answer it before you leave, we would appreciate your responses. And I'll just mention that next month's Lunch and Learn presentation will be on October 21st, and our presenters will be Catherine Kalasa and Tova Wolf, who will discuss Should I Go Mediterranean? Evidence-based dietary strategies to reduce pain and improve function and quality of life. I will send out a link to that registration in the email later this week. Thank you so much for joining us today, and thank you, Dr. Lascelles.